In ovo, electroporation begins with opening the egg to expose the chick embryo at Hamburger-Hamilton stages 9 to 11. DNA is injected into the midbrain region. Then, electrodes are placed left and right of the midbrain and an electrical voltage is applied. This causes the neural progenitor cells at the ventricular zone to take up the DNA. For ventral transfection, one electrode, the anode, is positioned under the membrane covering the yolk below the midbrain area. The cathode is placed laterodorsally at the opposite side of the midbrain, before an electrical voltage is applied. Please take care that the electrodes do not contact the neural tube. 24 hours later, at Hamburger-Hamilton stage 16 or 17, the embryos are removed from the egg, fixed, and analyzed by antibody staining or in situ hybridization. GFP-positive cells indicate the area of gene transfection.
Electroporation of microRNA precursors molecules or a microRNA duplex provides an immediate but short effect. To assure a longer expression of microRNAs, we electroporate an expression vector that contains sequences coding for microRNA together with a vector expressing GFP to visualize transfected cells.
MicroRNA expressed from a vector will be present earliest 4 hours after electroporation. Please see our written protocol for detailed instructions regarding sterilization of tools and preparing solutions and constructs we used.
Hello everybody, I am an MD student in the